Hello, gentlemen. As I said on Friday, because next week we don't have enough time in class to talk about, to have this kind of lecture, we do need to have a lecture about the control of the cell cycle and the checkpoints inside that cell cycle before we actually go to lab. It is important that we have this lecture before lab so that when we're looking at cells that have divided, we can understand chemically, biochemically, and molecularly what's going on so that we can further appreciate the process and be able to analyze malfunctions to that process, and we can to better understand that process in general. So we're talking about the control of the cell cycle. By now, you should have watched that flash video that I sent you that discusses the different kinds of checkpoints that prevent or that make sure the cell is ready to enter new stages in its lifespan. That video described three kinds of checkpoints. There's the G1 checkpoint, the G2 checkpoint, and the M phase checkpoint, as we're writing on our screen here. All right. The G1 checkpoint makes sure that the cell is ready to duplicate DNA. Duplicate DNA. I'm sorry if I'm writing illegibly. The G2 checkpoint makes sure the cell is big enough to divide. And then the M phase checkpoint makes sure that that cell's chromosomes are all lined up on the metaphase plate, because we don't want to divide a cell and have unequal amount of genetic material in the two new daughter cells, because those would not then be exact replicas. They wouldn't be identical cells. Okay, and the video actually focused specifically on our M phase. So let's take. I'm going to circle that here. Okay, our M phase, and we're going to take a closer look at what's going on in M phase, at the M phase checkpoint. So let's assume the cell has gotten past the G1 and the G2 checkpoints. And the cell is now prepared to divide. It's entering mitosis. Let's understand chemically what's going on. We've understood so far what's going on with chromosomes, but what's causing those chromosomes to act the way they do? So the question is, how does mitosis happen? How does cell division or nuclear cell division happen chemically, molecularly? What chemicals, what molecules are involved? Well, there are a few of them. Let's talk about two in particular, two molecules. First one is going to be called cyclin. Okay, cyclin. Cyclin is an enormously important molecule. It's an activator molecule. Right? Cyclin activates an important enzyme called CDK. Right? CDK, I'm going to write over here, is short for cyclin dependent kinase. There we are again. Look at that. Right, kinase. We know what kinases do now. If you remember from last unit, kinases phosphorylate other proteins. Right, they usually activate proteins. Okay. So, and as you can see here, cyclin-dependent kinase is dependent on cyclin. So as you can see, according to this diagram here, is we've got CDK and we've got cyclin. And what happens, as you can see, is they fit very well together. Cyclin is an activator for cyclin-dependent kinase, for CDK. And when cyclin binds to CDK, it activates the kinase activity. And now we no longer call it cyclin and CDK. We start calling that complex MPF, which stands for the mitosis promoting factor. All right, usually as the name implies, or you can tell what the molecule or that complex does based on its name. It promotes mitosis. The mitosis promoting factor has a number of activities. All right, as you can see below here, the MPF enters the nucleus, all right, and initiates spindle assembly. It initiates the breakdown of the nuclear envelope. It stops all gene transcription, and it starts condensing chromosomes. All right, as you can see, that's a lot of what happens in prophase and prometaphase of mitosis. So MPF, the mitosis promoting factor, which is a combination of cyclin and CDK, all of those activities are generated or initiated by MPF. Okay, so molecularly, how does prophase actually work? Well, it's the result of the transcription factors, that is to say, for MPF. Okay, as you can also see, according to this diagram, it goes cyclin and CDK and MPF go through phases around the cell cycle. All right, as the cell goes through its G1, S, and G2 phase, cyclin starts building up. We increase cyclin as we go through these phases. All right, as we approach the G2 phase and approach mitosis, cyclin is continuing to bind to CDK. Once it reaches a critical threshold value, all right, the mitosis promoting factor, the kinase activity of CDK, starts activating all of these actions. And then as the cell leaves mitosis, CDK, or cyclin is degraded, and CDK is no longer activated. And we have to wait for more cyclin to build up. Okay, that's what we see here. Let's take a look at a different kind of diagram that shows us similar information. Okay, so here we are at this diagram. We see the relative concentrations of our important molecules, cyclin in particular, and then cyclin bound to CDK to generate activity, the MPF activity. So what you can see here is as cyclin is increasing in concentration in the cell, as it reaches a critical threshold value, the kinase activity in CDK shoots straight upward, all right, and then it's very soon thereafter decreases in activity. All right, but by that point, mitosis has happened. All right, the kinase activity has decreased, cyclin levels have dropped dramatically, and we're ready to build cyclin up again. So it's a continual cycle. Right? It's a cell cycle, which is why we have names like cyclin, and we talk about the cell cycle. Right? This is a cycle or a cyclic activity where throughout a cell's life cycle. Okay, so rising cyclin levels cause activation of MPF, triggering cell division. And while the mitosis promoting factor, MPF, while it has activity like condensing chromosomes and breaking down the nuclear envelope and stopping gene transcription, it has all of those activities. It has another very important activity. All right, mitosis promoting factor activates this new complex of proteins called anaphase promoting complex, APC. Sometimes you'll see it as APCC because it has another name that we don't need to worry about right now. Okay, so let's talk about anaphase promoting complex. However, before we can understand the true nature of the APC, or the anaphase promoting complex, all right, we have to understand what's going on before anaphase. All right, why do we need a complex to promote anaphase? What's preventing anaphase from happening anyway? All right, so we have to take a look and remember that we've got at metaphase we've got chromosomes, okay, being attached to spindle fibers. Right? Those spindle fibers are actually pulling already, but those chromosomes don't separate. Why not? All right, so we take a look. Remember that the spindle fibers are pulling on the sister chromatids, but those sister chromatids don't separate until anaphase. Well, why not? Let's take a look. Well, as you may have guessed, right, usually in the cell, if there is a process that needs to take place, it usually involves proteins and enzymes and all that sort of stuff. All right, so the reason why those chromosomes or those chromatids don't separate until anaphase is because there's a new protein associated with them. Well, there has been a protein associated with them. All right, that protein is known as cohesin. Cohesin acts as a cohesive element. All right, it acts to bond those two chromatids together, and it doesn't break apart until signaled otherwise. So those chromatids won't break apart until cohesin has been broken down or degraded. All right, well, how do we do that? Well, we use another protein, or another enzyme rather, known as separase. But separase isn't active, all right? Separase isn't activated until it is until it is disassociated with another protein called securin. All right, so originally there's this protein complex called the securin separase complex. All right, if you remember in the flash video, right? First of all, those hoops that they were talking about that hold those chromosomes together, those chromatids together, those hoops refer to cohesin. Right? And those scissors that break or that snip those hoops, that would be separase. However, there is a sort of a security latch or a safety on those scissors kind of molecules. There's a safety on that separase. That safety is called securin. It prevents or it inhibits separase activity. All right, so we've got a bunch of different proteins and molecules preventing and wanting to start start breaking down and separating those chromatids. So here comes along APC. Okay, and APC, what it does is it triggers the degradation, right, the elimination of securin from that securin separase complex. So now all we have left is separase, and separase uses its scissors activity to break apart cohesin. When cohesin is broken apart, right, those spindle fibers that had been tugging and pulling on the chromatids are successful now at breaking them apart, and anaphase happens. And anaphase happens, and telophase happens, and so on and so forth. Okay, so we have our APC is anaphase promoting complex. The APC activates separase by releasing securin, and separase degrades cohesin, allowing the chromatids to separate from the pulling of the spindle fibers. Here we see a similar kind of image, but it actually demonstrates the separase securin or the securin separase complex over here. We can see that the active site of separase is over here, but securin is securing it from actually working. Our APC is going to get rid of securin, degrade it, right? And separase is going to act on our cohesin molecules. All right, here's our cohesin molecules. We get rid of them, we break them apart, and our chromatids separate. And as you can see now, this is the control of the cell cycle, the molecular control. All right, we have nothing can happen until checkpoints have been reached. We can't move on to anaphase until the APC has been activated. We can't move on to we can't activate the APC unless we have mitosis promoting factors, and we can't activate the mitosis promoting factors until appropriate levels of cyclin have been generated. All right, so we talk about checkpoints. All right, we talk about all of this control. There are many control mechanisms, and now you know them.